In part 4 of this lecture, we'll talk about the role of these macromolecules in phage biology. Right, so one of the first things we talked about when we brought up chemistry are these emergent properties. Right, we have all of these macromolecules. They are made up primarily of those 6 carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur atoms. So those are the primary building blocks of all cells and all organisms. Right? But you learned how those atoms come together to form like these complex, interesting molecules. And those molecules, because of the interactions between those different atoms, have unique chemical properties. Right? Sometimes there are different chemical properties within the same molecule. Those molecules then to come together to form chemical reactions with each other, other chemical interactions, and form the structure and the functional parts of cells and organisms. So here we're going to discuss 3 examples of how these molecules play functional roles in biological systems. Right? How those complex chemicals make up bacterial cell membranes and cell walls, how the complex structure of bacteriophages allows them to penetrate the walls. And then we'll talk about how we use our understanding of these chemicals in order to reveal some of the functions in biology. So remember, we previously talked about our bacteriophage, our hosts for our phages, the bacteria. Those bacteria have what seems like a relatively simple structure. They have a cytoplasmic membrane that contains the stuff on the inside, including the cytoplasm, the ribosomes, and the bacterial DNA. And then they have a rigid cell wall associated with that cytoplasmic membrane. But also remember that this structure of the bacteria is more complex than it appears. It's made up of a complex mixture of these molecules that form these interesting properties of the bacteria. So again, the bacterial cell membrane, like all membranes, is made up of these phospholipids. It's made up of this phospholipid bilayer, where you'll recall these phospholipids have these hydrocarbon chains that are very hydrophobic and don't like to interact with water. And then they have these charged phosphate ends that are hydrophilic and do like to interact with water. Now, life occurs in water. And so on the outside and the inside of the cell, there is water. But the hydrophilic ends of the phosphates interact with the water on the outside and the inside of the cell. And then the tails of the hydrocarbons form a hydrophobic barrier between the water outside the cell and the water inside the cell. Right? So this hydrophobic barrier prevents many molecules that are dissolved in water from moving outside to inside the cell and vice versa. And this allows cells and organisms to carefully regulate what goes into and out of their cells. And this is an important property of life. Now, the membrane doesn't just contain phospholipids. It's actually a complex mixture of lipids, proteins, and carbohydrates. There are proteins associated that float in that semi-liquid membrane that are either associated with one side or the other of the membrane. And there are also proteins that span from the inside to the outside of the cell. Some of these proteins include channels that selectively allow certain molecules into or out of the cell. Many of the proteins on the outside of the cell membrane also have carbohydrates attached to them. We call these glycoproteins. So they are proteins that are modified with a carbohydrate chain. So bacteria are generally grouped into gram-positive type bacteria and gram-negative type bacteria, and these are 2 distinct evolutionary related groups. They are distinct structurally in the structure of their cell wall and cell membrane. So both of these bacteria have phospholipid bilayer membranes. And then they have a cell wall, which is a rigid structure composed of a chemical called peptidoglycan. So in gram-positive cells, we have a thick layer of peptidoglycan around a single phospholipid bilayer membrane. And in gram-negative cells, we have a thin cell wall that's between two phospholipid bilayer membranes. We have the outer membrane and the inner membrane. So again, gram-positive cells have this thick layer of peptidoglycan around a single phospholipid bilayer membrane. Right? In that membrane, there are proteins and other things. Then in the cell wall, which is made up the peptidoglycan, the cell wall has these several layers that are layered together, joined together covalently by teichoic acids. So these are molecules, they are carbohydrates, and they form covalent linkages between the layers of peptidoglycan. Right? And then the teichoic acids, some of those teichoic acids have a lipid attached to them and allows the cell wall to be anchored to the phospholipid bilayer membrane. Gram-positive cells include the actinobacteria, such as M. foliorum, which is the host that we've been using in the phage lab. Gram-negative cells, which also contain many important phage hosts, they have a different structure. Again, they have a single layer of peptidoglycan, a thin cell wall, and that's between two phospholipid bilayer membranes. Again, those membranes have proteins and other things in them. The space between those two membranes we call the periplasmic space, and in that periplasmic space is that peptidoglycan cell wall. The outer membrane of gram-negative cells has a number of important chemicals. Importantly, it includes lipopolysaccharide or LPS, which plays a role in the interaction of the gram-negative cells with our immune systems. Gram-negative cells include many important bacteria, including E. coli and Salmonella. So, what is this peptidoglycan that makes up the cell wall? So, peptidoglycan is a complex polymer that's made up of polymers of carbohydrates, where those those polysaccharides are made up of repeating disaccharide units. And the sugar that makes up the disaccharide is called NAM, NAG. Right? So these are these two sugar monosaccharides that join together to make a disaccharide, and then are joined to make these chains of carbohydrates that are shown here in the balls in this peptidoglycan cartoon. Then there is a amino acid linker between those chains of carbohydrates. So those amino acids are covalently bonded together, and they form a chain between those layers of carbohydrate polymers. Right? So just like we talked about the structure of cellulose, we have these chains of amino acids that are covalently bonded together, and then we have these amino acid chains that are linking together the layers through additional covalent bonds. So this creates a extremely strong mesh structure. It's a rigid structure that protects the bacteria from a number of hazards, including bacteriophage. Okay? So phages have to penetrate the cell wall in order to get their DNA into the cell. And so they have to use force to do this. So to understand how we know how phages get into the cells, we need to know a little bit about how we learn about the structure of these biological molecules. Right? So we have several biophysical techniques that we learn the structure of these biological macromolecules using. Okay? We have X-ray crystallography, where we have a beam of X-rays. And X-rays, when we shine them through a complex macromolecule like a protein, the electron cloud that is associated with the atoms bends the X-ray beams. And we can look at the diffraction pattern that's formed by shining an X-ray through a crystallized protein in order to learn about where those electrons and where the density of the atoms are in the crystal. And from that, we can use complex computer algorithms to map all the individual amino acids of the protein or nucleic acids in the or nucleotides in the nucleic acids to form a three-dimensional structure map. We also use nuclear magnetic resonance, where we look at the interactions between atoms in these macromolecules by applying a super strong magnetic field. This causes a different resonance pulse to be produced based on the way those atoms are bonded to each other. And this can tell us a lot about how the atoms not only are bonded, but are moving relative to each other. And it tells us something about the dynamics, or the way the molecules move. Unfortunately, because of the super strong magnets needed, we can really only study small proteins or nucleic acids. Electron microscopy is another important technique that we use to understand the structure of molecules. Right? Electron microscopy uses electrons to image things that are very small. And it gives us high-resolution images of small things like bacteria and viruses. Right? But electron microscopy is a high-powered microscope, but it's not quite powerful enough to really see detail of things as small as individual proteins or nucleic acids. However, in recent years, cryo-electron microscopy, where we use a supercooled solution or a supercooled sample of proteins or nucleic acids that are spread on a grid, to take millions of individual electron microscopy images, and then using a computer to take all those different images of individual molecules that should be relatively the same to create a density map where we can increasingly look at these molecules in higher and higher resolution. So to give you a sense of how we use these techniques to learn about the structure of important macromolecules, I should tell you the story about Photo-51, the discovery of the double helix. Okay, so Watson and Crick get credit for learning the structure of the double helix. But it was a scientist named Rosalind Franklin that really provided the key scientific evidence to understanding the double helix structure. So Rosalind Franklin was an X-ray crystallographer. She had been crystallizing various forms of nucleic acids and taking images of them by shining an electron beam through that crystal. Rosalind Franklin produced a very nice high-resolution image of the X-ray diffraction pattern by shining X-ray beam through a crystal of DNA. Right? That X-ray pattern showed this characteristic X, which was consistent with the helical turns in the double helix. Right? So by looking at the X diffraction pattern, Watson and Crick, when shown a copy of Franklin's data, they knew that the structure of the nucleic acids had to be helical. They then built these metal models of the atoms as they were associated in the nucleotides that they knew the basic monomer structure of. And then they were able to figure out how those puzzle pieces could fit together to form a helix where there were complementary hydrogen bonds between the individual nucleic acids. And that's why we know the structure of DNA is in this double helix. So, back to bacteriophages. Bacteriophages have this problem. They have to penetrate the rigid cell wall of bacteria in order to get inside. And different phages have different structures for penetrating that cell wall. So remember, we have these three different groups of bacteriophages. We have the Myoviridae that have the contractile tail. So their tail consists of many proteins that are able to squeeze together like a syringe to inject its DNA genome through the cell membrane in the cell wall. The Siphoviridae have these long, flexible tails, and their tails consist of protein subunits that are able to flex back and forth, and the flexing motion allows the DNA to be moved through the tail and into the cell. Then the Podoviridae, they have these short stubby tails that have these incredibly powerful protein motors on the end of that tail that move or force the DNA into the cell. So how do we know how these tails work? Well, we know based on biochemical and biophysical data. For example, here are some recent pictures from a study done using cryo-electron microscopy to look at the structure of T4 bacteriophage as it injects DNA into the cell. Okay. So these scientists made what are called E. coli mini cells, where they make E. coli, instead of going through regular cell division, make these little lipid droplet buds. And because those buds contain the receptors for the T4 bacteriophage, the bacteriophage can latch on to those mini cells and will try to inject its DNA into the mini cell, even though it's not a full functioning cell. So the scientists made bunches of these mini cells and then infected them with T4 bacteriophage, and then took millions of these electron microscope images of phages, injecting their DNA at various points. You can see the phages as they attach to the cell, and then as that tail contracts shorter and shorter, and you can see even in these pictures the DNA double helix injected into the cell through the cell wall and the cell membrane. At the end of this process, where we see the inside of the phage capsid no longer has the density associated with the DNA genome. And in fact, you can see the inside of the capsid becomes less dense as more DNA is injected into the cell. This is the end of the process. And so the scientists can look at their millions of individual images and put together a time lapse of how the individual proteins in this bacteriophage function in injecting that DNA through the cell. So this is a relatively complex experiment. But let's talk about an earlier one where scientists also use their understanding of the chemistry of nucleic acid and proteins to make an incredibly important discovery. So Martha Hershey and Alfred Chase were two scientists, pictured here in 1953. Right? They knew at the time they did this experiment that phages were made of both protein and DNA. Now, they also knew that phages reproduced by infecting bacteria. And so if you mixed phages and bacteria, there would be new phages made containing protein and DNA that you could isolate from those batches. However, in the early 50s, there was an important scientific controversy that scientists didn't actually know which chemical, protein or DNA, was responsible for initiating the process of making new phages. Which chemical was the genetic material? So Hershey and Chase wanted to figure out which one of these chemicals was responsible. And they knew an important fact about the difference between proteins and nucleic acids. Right? They knew that nucleic acids were contain these ribo and nitrogen base parts that consists of carbon, hydrogen, nitrogen, and oxygen. But they also know that the phosphodiester bonds that form the backbone of these nucleic acids contain phosphorus. They knew that amino acids that make up proteins are made of carbon, hydrogen, oxygen, and nitrogen, just like nucleic acids. But they also knew that two of these amino acids contain the atom sulfur. Right? So they used radioactive phosphorus, or P32, and radioactive sulfur to specifically label either nucleic acids or amino acids in their preparations of phages. Okay. So in their experiment, they had two different batches of phages. One they grew with radioactive sulfur, right? And those were phages where only the radiation was found, but only in the amino acid containing chemicals in the phages. Right? Then they had another batch that they grew with radioactive phosphorus. And so those phages had radioactivity only in the nucleic acid portions of the phage. Right? So they then took their bacteria, and so they had these two experimental groups. So some of the bacteria they infected with sulfur-containing phages, and then another patch of bacteria they infected with phosphorus labeled phages. Right? They allowed the bacteria and phages to mix to start the infection process. Then they briefly blended the phage and the bacteria. Now remember the lytic cycle in infection, where the phages attach to the host. Right? So that takes a little time. And then they do something to the bacteria. And then the blending shakes up the phages and the bacteria so that the attached phages will come off. Then they immediately centrifuge that solution of bacteria and phages. And centrifugation pulls heavy things to the bottom, so the bacteria are found at the bottom of the tube. And then in the liquid upper portion, that contains the remaining phages. So this is the data from Hershey and Chase's experiment, and I wanted to briefly walk you through how to think about this data before we interpret. So we have, in the first column here, we have the running time in the blender. So these phages were either blended for 0 minutes or 2.5 minutes just prior to the centrifugation step. Next, they have the multiplicity of infection. And so here, they either use 0.6 phages per bacteria, so there's less phages than there are bacteria, or they use 6 times as many phages as bacteria. Then they have their the results of the two experimental groups. They have their infections that were done with P32 labeled phages, and they can measure the amount of the radioactivity that was eluted or remained in that liquid portion after centrifugation. And then they have their sulfur labeled phages, and again they can measure the amount of isotope that was eluted, that remained in the liquid portion after the centrifugation step. And so here's the setup of their experiment. They have their P32 labeled phages. They have their S35 labeled phages. Those phages they allow to incubate with the bacteria, and the phages presumably attach to the bacteria, and then they do something to the bacteria. So that even if you remove the phages at this point, the bacteria produce new phages when you incubate them in a tube. Right? So they incubated these radioactive phages. They blended and centrifuged so that the bacteria pelleted to the bottom, right? And that the phage shells remained in the supernatant. They can then look at the presence of radiation in the bacteria, or in the figure that we'll look at, they can look at the presence of radiation in this liquid portion that contains the phages after they started the infection in the bacteria. And so here's that data again. Again, this is 0 minutes of blending and 2.5 minutes of blending. So whether or not they did not blend or they blended for 2.5 minutes before centrifugation. So if they incubated their phosphorus labeled phages with and didn't blend them and centrifuged, the phages would stick to the bacteria. So only 10% of the radiation remained in that liquid part, and most of it pelleted to the bottom with the bacteria. Then, if they subjected those phages to 2.5 minutes of blending, then some of that radiation went back into the supernatant. So without blending, they found 10% of the radiation in the supernatant. With blending, they found 21%. They did the same thing with their sulfur-labeled phages. They incubated the phages, and if they didn't blend it, pelleted the bacteria, only 16% of the radiation was found in the supernatant, in the liquid portion, and the rest was pelleted with the bacteria. But then, if they blended for 2.5 minutes after initiating this infection, they found 81% of their radiation came back into the liquid portion even after centrifugation. So presumably most of the radiation for the phosphorus-labeled stuff went to the bottom of the tube and stayed with the bacteria. And then most of the sulfur-labeled stuff remained in the liquid portion and did not go with the bacteria. So let's go back to our experimental setup. Remember, they incubated these phages, blended or didn't blend, and then pelleted. When they blended with their P32 labeled phages, they found most of the radiation remained with the bacteria in the bottom of the pellet. When they used their S35 labeled phages, they found most of the radiation remained in the supernatant. Right? So, what do these results say about which chemical, DNA or protein, was the chemical that was injected into the phages in order to start the reproduction of new phages in the bacteria? DNA or protein?